Hey there! We're going to do a quick video here on transferring energy in cells, like in cell bioenergetic type reactions. This is a sort of connection between the idea that there's different types of energy and to how do cells harness this energy and make use of it. So, in a previous video, I discuss a whole bunch of different types of energy, relate them to kinetic and potential energy. So there's the energy of things moving and the energy, of course, that is stored. And a big part of biology is finding ways to take stored energy in one form and turn it into stored energy in another form while losing as little as possible to the external processes of heat and entropy and all of that second law of thermodynamics stuff. To do that, there's a lot of different transfers that happen, and we use kinetic energy in a lot of interesting ways to really follow how this works. But there's four main things in biology that you're going to see in terms of energetics. A big one, of course, is light. All of the energy on this planet really starts as the kinetic energy of light. Photons are coming down from the sun. Something has to capture that energy and turn it into something else. And the plants do that. We'll get back to plants at some point because we'll talk all about photosynthesis. So that piece will come back. The next of these forms is the one that is related to the part we're most interested in, which is the chemical bonds. So we are super interested in making chemical bonds and storing our energy, preferably in forms that are then easy to use later. To do that, we have to make and break chemical bonds in a variety of ways, and preferably in ways that have the most, have the least loss as possible. So we're trying to conserve as much as we can. Chemical bonds are strongly related to electrons. And so what we're really talking about here is we're talking about moving electrons in some way. A lot of times we try to squish molecules close together so they move more easily, or we attach them to something that gives them an electronegativity that makes them push further apart. But either way, we're trying to deal with the movement of electrons. The kinetic energy form that this is involved in is electrical energy, because this is the movement of electrons, or in this case, breaking and making of bonds. I'm going to get back to the electrical energy, but there is one more type that is important, and that is an energy that relates to position. So again, here's our potential energy, is that things live in a certain spot, but want, in terms of energetics, to move to another one. So a big part of the positioning and moving that happens in biological systems happens by a process called osmosis, in which water specifically, but other things as well, move to areas of low concentration from areas of high concentration. It is a tendency. They're going to go in that direction, mostly because there is a certain amount of entropy occurring. They tend to spread out, and they tend to move that way. And that sort of extra motion can get caught by other things and dragged along with it. So a lot of times osmosis is used as an energetic process. If something is moving with a gradient, it can pick up something else and move it in a different direction that requires energy. So that's a key thing. But let's get back to that electrical energy for a moment, because sometimes it's hard to nail electrical energy into chemical bonds. But you really have to think of it as the movement of electrons. The movement of electrons is a really specific biological concept. It is so specific, in fact, that it has its own name. We call it redox. Now, redox is not just the fun name of something, it's actually a portmanteau, which is where you take two words and shove them together to make one word. Look, linguistics lesson as well. And in this case, those two words are reduction and oxidation. So we have reduction, we have oxidation. These are both chemical reactions, and they have to work together because of the way electrons essentially work. If you have an electron over here, it's going to move over there, which means something loses one, something gains one, something is reduced, something is oxidized. Let's start with the really big picture of redox, which is really where the words come from. Starting with oxidation, which is the idea that oxidation is the addition of oxygen to something. Now, long before we really fully understood the way chemical bonds worked, if you added oxygen to something, it oftentimes changed it, and therefore it was oxidized. To add oxygen to something, it has to come from somewhere else, so something else is reduced by removing oxygen. Now, oxygen very rarely hangs out on its own. There are O2 oxygen versions, but really, oxygen tends to make connections with other atoms, and specifically with hydrogen, for example, in the form of water, which is one of the most valuable and immense subjects of biology and on our planet. Because of the way oxidation reduction works, if oxidation is adding oxygen, reduction is adding hydrogen. And as before, if reduction is adding hydrogen, oxidation is often deemed to be removing hydrogen. So these are the large-scale versions of reduction and oxidation. But in dealing with them, they're really happening on an electron-based level. So we should understand what they are in terms of electrons. So oxidation is when you actually take away electrons. Now, in a lot of ways, this kind of makes sense because as you're adding and subtracting these different oxygens and hydrogens, you are creating varying kinds of chemical bonds. By adding oxygen to something, you're typically using up or connecting two electrons, so losing electrons makes sense. So you would have less electrons, which I am going to denote as a tiny e, the negative sign. Well, maybe I won't. Less electrons. And of course, if something has less electrons and that's oxidation, guess what reduction is going to be? It's going to be more electrons. And so further, one more way to keep track of how these work, because this will become important, is that if oxidation has less electrons, which have a negative charge, oxidized things are going to be more positive, and therefore reduced things are going to be more negative. So these are other ways to follow reduction and oxidation. So this is how you know anything electrical is happening in biological reactions, is you see any one of these things coupled with the opposite. So you see oxygen moving from one place to another, hydrogen moving from one place to another, more and less electrons or changes in charge. That's how you know there's redox. How do you keep track of which one is which? I have a neat trick for that. On the oxidation side, it is less electrons. We often say you lose electrons. So if you have lose electrons for oxidation, what you have is Leo. Now on this side, you actually pick up electrons. You have more of them, so you gain electrons. And this is reduction, which is grr. And if I appropriately Leo lose electrons and grr gain electrons, what I might have, just to help me out here, is my good friend. I promise this drawing will get a little bit better. Not much, though. Leo the lion, who says grr. I'll give Leo some eyes and a little nose and one of those kind of funny animal smiles. There we go. Look, I made a lion. Leo the lion says grr. Losing electrons for oxidation, gaining electrons for reduction. So anytime you are following along in an enzymatic or chemical reaction, these are the things you're looking for redox, and you can remember what direction is which by saying Leo the lion says grr, which honestly I still say every time I deal with redox, and I have been studying this now for almost 20 years. So hope you enjoy that.